In additional, microbiology from Tribhuvan University, Nepal. Dr. Thapa taught microbiology course at various colleges in Nepal, including Central Department of Microbiology for over 8 years. He is also visiting associate professor at Central Campus of Technology in Taran. His research primarily focuses on micro from ecology to clinical application. He has published over 25 peer-reviewed articles and edited 3 books. He is currently involved in organizing conference seminar in Nepal and abroad. Since 2012, he has been organizing the conference series International Conference on Infectious Disease and Nanomedicine in Nepal in collaboration with societies, including the American Society of Microbiology, European Society for Clinical Microbiology and Infectious Disease, and the Chinese Academy of Science. Before we start into Dr. Santosh Thapa's presentation, I want to say a few housekeeping rules. First, everyone should be muted, and at the end of the presentation, we'll have a question and answer session. At that time, you can ask the question or type in the question. This seminar is being recorded for educational purpose and is being in Facebook of PRIPS and RIPS Facebook page. So, Dr. Santosh Thapa, you can take the stage.
All right. Thank you very much, guys, for inviting me. Thank you very much for a nice introduction. I really appreciate that. Let me share my screen and we'll talk about it. Oh, you disable participant screen sharing. So is there a way I can share my screen, or because it says the host disabled? Okay, got it.
No.
Yeah, let me try again. Yep, looks like it's working. Were you guys able to see my screen?
Yeah.
Okay, good. So once again, thank you guys for inviting me. As you already pointed out, I have a background in microbiology with the training in molecular genetics and then microbiome sciences. Over the last 7 years or so, I have been actively involved in researching in microbiome science, particularly human microbiome, also the various microbiomes of the insects, including various ticks and other disease vectors. So today, the title of my talk is the Deciphering Clinical Utility of the Human Microbiome. As the announcement already made, I am affiliated with the Baylor College of Medicine and Texas Children's Hospital in Houston, Texas. My primary research focus on the microbiome stuffs, whether it be human samples or whether it be the other animal samples. So this is the brief outline of my presentation today. I'll be talking about the basic concept about microbiome, and then much more, little bit about the human microbiome. And then I will dive into the microbiome in health and disease. Then my discussion will be focused on how the microenvironmental factors such as diet or medications can impact the microbiome in health and disease. And then I'll be talking very briefly about the fecal microbiome transplantation study that we performed here at the Texas Children's Hospital and then Baylor College of Medicine. And then finally we'll summarize the overall talk. So many of you have already heard about it, microbiome and microbiota in the literature. If you look at it, you may find that people use interchangeably as a microbiome and microbiota. But in 2020, the European Framework for Microbiome, led by Dr. Berg et al., published a very nice study in the journal Microbiome, where it's clearly laid out about what the microbiota and then microbiome are. In general, when you say microbiome, it's the collection of the microorganisms in a particular ecological environment or particular sample, for example. So when you talk about the microbiome, it is the combination of the collection of microorganisms plus the theater of activities. When I say theater of activities, it includes not only the microbiome, or community of microbes, but also their structural element, including nucleic acids, lipids, and polysaccharides, as well as the microbial metabolites like signaling molecules or toxins or other organic molecules. And not to forget is the environmental conditions. So this is the much more recent concept of the microbiome, which includes all the collection of microbes in a particular ecological unit, plus their activities, which is the the signaling molecules and then other toxins, and as well as the environmental interactions. So, and there are various methods in the literatures where you study the host microbiome interactions at different molecular levels. The basic one is the DNA-based approach. It's just to see who are there and what they can do. And the most widely used techniques for studying the host microbiome interactions based on the DNA, probably you guys have heard a lot about, is the 16S ribosomal RNA sequencing for bacterial microbiome analysis. And similarly, 18S and ITS for fungal sequencing. On the other side, there is the shotgun metagenomics, which is the sequencing the whole genome present in a particular sample. So there are two things in the DNA-based approach. One is the marker gene-based sequencing, which we popularly call as amplicon sequencing, because we amplify their PCR fragment, and then we do sequence them. And that's the best example would be the 16S ribosomal RNA sequencing. And then shotgun metagenomics, as the name indicates, it includes the whole or the metagenome, the complete metagenome that is present in a particular sample. And it includes not only the bacteria, but also the fungus, other, the archaea, and then you can also the viral part as well, or bacteriophages, if you will, in the human microbiome. And the next approach is the RNA-based approach, which basically looking at how do they respond to certain environmental stimulus, for example, or stress or antibiotics treatment, and then what pathways are activated during the process. It is popularly called as metatranscriptomics. Similarly, the protein-based approach as metaproteomics can be used to understand how they are interacting with their host, or how what proteins are being produced, things like that. It's pretty much based on the mass spectrometry-based technique. And similarly, there are other approaches we call them metabolomics, which is the metabolites-based approach. Basically, what chemicals are produced because of let's say the gut bacterial microbiome interactions or their activity. And then we use mostly the mass spectrometry-based LC-MS or MS for this purpose. And for this talk, I'll be focusing mostly on the DNA-based approach, particularly the 16S ribosomal RNA sequencing, and a little bit about the metabolomics approach that we take into account for our studies. So just to give you a brief ideas, one of the most popular technique in the microbiome field is the 16S ribosomal RNA sequencing. You may probably heard about the 16S ribosomal RNAs involved in protein synthesis, the smallest unit in the prokaryotic ribosomal RNA gene. The 16S ribosomal RNA is quite conserved in the bacteria, so that's why we've been using it since a long time. Structurally, the 16S ribosomal RNA contains the gene with the variable and then constant regions, basically. So the B stands for the variable regions. Some call it as the hypervariable region as well. So there are B1 to B9, hypervariable regions, about 1,500 base pair, which is interspersed between the constant regions. And then once you choose the region of particular interest, for example, let's say B1 to B2 or B3 to B4, then you amplify with a specific primer doing PCR. Then it has the barcode and sequencing with the index PCR. Then you move on to the sequencing, for example, in the Illumina MiSeq, popularly used in these days. And then once you get the data output from the machine, then you would use the whole bunch of bioinformatics approach to analyze the data. And the most widely used technique of the platform, if you will, is the CHIME, which is the quantitative insight in microbiome ecology, as well as the other softwares like Mother and other. There are various softwares that are widely used in the microbiome field. So long story short, the 16S ribosomal RNA is most widely used in the field of microbiome, just to understand the composition and then what is different between the two ecological samples or two different sample types, whether it be healthy subjects or the disease subjects. This is just a brief introduction. I'm not going to go in details about it, just the idea of one of the techniques that I'll be talking about today. In overall, you can also do the, like I mentioned before, shotgun metagenomics for this purpose as well. So let's talk about a little bit on the human microbiome side. I know many of you have heard about the human microbiome, its implication in various diseases and various, various axes, whether it be gut-brain axis or gut-liver axis or gut-lung axis, and so on and so forth. So what basically is the human microbiome? As we all know that this is the picture from our upcoming book chapter, the textbook of pediatric infectious disease, 9th edition, is from Elsevier, which is basically showing that the larger pie chart shown here for different anatomical sites are for bacteria, and the smallest, the green one with the little one is the fungus, and the one with the much more light blue is the prophase or the viruses, basically bacteriophages, if you will. So looking at the different anatomical site, there are the composition of the microbiome varies based on their anatomical location. So across the human body, there are estimated to be around 40 trillion microbes per person, who is equivalent to 1 to 3% of a person's body weight. And if you convert to the numbers, the much more recent estimate of the ratio of the bacteria to human cell is about 1.3 to 1 to 2.3 to 1, according to Santosh et al. In 2016 study. So the human microbiome is much more diverse and much widely studied in the gastrointestinal system, or we call it gut microbiome. And then skin microbiome is also widely studied. The less studied are other microbiomes, such as the upper airway microbiome or respiratory microbiome and other body parts. This picture shown here is a very nice layout by the Gabriela et al. Published in last year, where you can see that the gut microbiome dysbiosis has a link to various diseases and disorders. As you all know that by now that the gut microbiome has been implicated in the right functioning of the various organs of the body, including the brain, including the liver, including heart or skin, and other organs. So there's when I say the dysbiosis, is sort of the imbalance, or if you will, you may call it as a there's a lack of homeostasis between the commensal microorganisms and pathogenic microorganisms, for example. So when there is a dysbiosis in the gut microbiome, there are various diseases that have been implicated because of that. The most widely for the the gut-brain axis is psychiatric disorders. We published papers in the major depressive disorders 2 years ago, and we are also working on the biggest study in the U.S. is the autism spectrum disorders and various neurological disorders. I'll be talking about a little bit about one of the neurological disorders that we published back in 2021, I believe. And then there are some gut-lung axis as well. It could be in asthma, or it could be in COPD, or in lung cancer, or it could be in the cystic fibrosis patients. And in terms of metabolism, it could be in cancer, which is a very very important field in cancer microbiome these days. Is much more emerging and recently in terms of how you can manipulate the microbiome for the responsive treatment of the anti-cancer drugs. So. And similar to the skin microbiome as well. So long story short, the gut microbiome dysbiosis has been implicated in a lot of diseases, and then the dysbiosis has been sort of checkpoint inhibitors. A checkpoint for the for management of the various diseases. Again, coming back to the definition of microbiome, it makes sense that when you talk about the microbiome, these are not only the live microorganisms present in a particular community, but also the theater of activities, quote unquote. So including the environmental conditions. So when you start working on the microbiome, or you're already in the microbiome field, then you need to think about not only what they are present there, what they're actually doing, how they're interacting with the environment, whether the environment could be the external environment or could be the host, or any things that are environmental in the sense. So one of the clinical utility of the microbiome in various fields of the medicine is the differentiating between the healthy and the disease state for a particular cohort, for example. So when I say the differentiation, it means that based on the microbiome composition, you may be able to differentiate between the healthy and the disease condition. And at the same time, you might be asking questions: what are the normal microbiome? What are the healthy microbiome like? So there's a big debate in the field. So what constitutes normal is not known. So basically what we take into account is that for a particular cohort having the same, age, same gender, and then same from the same geography, we take into account of those samples as the healthy control, and then compare with the disease person or disease individual. So there may be, differentiation, or what we call the heterogeneity between the healthy individuals, and the similar is true for the disease person. But there is consideration that if you take into account of the age, or gender, or other confounding factors, then you would have a better chance to have the comparative evaluation of the microbiome between the healthy and the disease condition. So this is one of the studies that we published back in 2020, where we looked at the microbiome signatures in the neonates with the central line associated bloodstream infection at the Texas Children's Hospital in Houston. So what basically the study design was that we basically used a catheter biofilm microbiome with and without the Klebsiella infection. And then we, collect the samples, extract the DNA, and then using the PowerSwell kit, and then perform the 16S ribosomal RNA sequencing. I believe it's a B4 reason, and then run the CHIME2 analysis, and then you were able to see these visual pictures here. So here in the x-axis are the two different groups, and in the figure A is one is the uninfected catheter, and another one is infected catheter. And in the y-axis is shown is the numbers, which is the relative abundance, which runs from 0 to 100%, or you can also put it 0 to 1, which basically is the ratio between these different types of bacteria. The A is showing the phylum level taxonomy composition between the two groups, whereas B is showing the genus level bacterial composition between these two groups. And essentially showing that we were able to show that there are differentiation between the two types of microbiome depending on the infection status. One of the prominent bacteria that we saw the difference was the Bradyrhizobium, which was less in the infected catheter as compared to the uninfected catheter. Similarly, Proteus bacteria was higher in the infected catheter as compared to the non-infected or uninfected catheters. So essentially, showing that the microbiome profile of the infected catheter biofilm microbiome is distinct from the uninfected catheters, and published in the 2020 in the +1. Within the same study, we also look at the skin microbiome. The skin microbiome is the skin swabs with the Klebsiella neonates. What we found basically is that there's not much difference between the infected versus non-infected, but the Staphylococcus shown here in yellow is the predominant organism present in the particular samples. And then the uninfected one has much more of the other bacteria, including unclassified Pastorella, and then Gamella and other species of the Anaerococcus. So the take-home message from this one is studies that based on the microbiome profile, you can easily differentiate between these, the infected versus non-infected catheter biofilm microbiome. When I say easily, this is sort of a little bit different than the gut microbiome. Please bear with me because I will be talking about not only the gut microbiome, but all other human body microbiome as well, just to show you an example of the different ways to deal with the data set. So this is the particularity for the skin microbiome and then catheter biofilm microbiome. Moving forward, there's another study that we conducted also in published in 2020 in Scientific Reports, where we look at the race. Is there any racial differences between the breast tumor microbiome between the white non-Hispanic and black non-Hispanic individuals residing in certain part of the U.S. The idea behind this project is that we know that for sure the environmental and the host or genetic as well as the socioeconomic factors is greatly influence the breast cancer prevalence. So we don't know the what type of the microbiome disparity exists, if we will, in the breast cancer subtypes. For that reason, we asked the question that if there is a microbiome differences based on the races. So what we did here in this study is that we collected the samples from the breast tumor, and then we also input the matched normal tissue adjacent to the tumor, which we call the NAT samples. We collected both from the black non-Hispanic and then white non-Hispanic individuals. And then interestingly enough, we found that in the black non-Hispanic and then white non-Hispanic, when you compare the triple negative breast cancer type, And then alpha diversity, shown here in the y-axis, is the Shannon index, which is basically, when I say alpha diversity in microbiome, the alpha diversity, the diversity within the sample. And then there are multiple indices that you can use for measuring the alpha diversity. One of them is the most popular, widely used, I will say, is the Shannon index, or sometimes they call it Shannon-Wheeler index, or just the Shannon diversity index. Clearly shown here is that with the between the tumor and the normal tissue type, you would see the difference in the the diversity, or the Shannon diversity. It's higher in the normal cell compared to the the tumor in the black non-Hispanic, where while the opposite is true for the white non-Hispanic. It's showing that one of the evidence showing that there is racial differences between the the black non-Hispanic and then white non-Hispanic with regard to the triple negative breast cancer. These are in addition to that, we also look at the microbiome, how we can clinically apply to one of the neurological disorder. It's very rare neurological disorder. We call it Rett syndrome. Maybe you have not heard about it. It is the neurodevelopmental disorder, which is due to the loss of function of the P2 gene, which is the X-linked methyl CpG binding protein gene. It's very rare. It affects about 1 in 10,000 females. It's mostly in the females or the girls. And over 90% of the girls with the Rett syndrome, they develop the GI problem, and thereby affecting their quality of lives and their health as well. So what we wanted to look at that because of these complications or GI disturbances, they may suffer from malnutrition or growth failure and a lot of issues with the GI related issues. Within this particular group, what we wanted to see is that are there any differences within the the Rett syndrome group that may be causing because of the dysbiosis in the gut microbiome, whether you can phenotype them based on the disease severity, for example, or based on the clinical phenotype. So essentially, we were able to show that the shown here in this picture, in the x-axis, the CSS, which is called as a clinical severity score. In a very layman term, it is the clinical severity of the disease, how severe it is, depending on their multiple different scores, multiple different mutations. And shown here in the y-axis is the Fisher alpha diversity, which is one of the diversity index that we use often for measuring the alpha diversity in the microbiome. So it's clearly shown that within the among the 3 groups of the disease severity, the more severe the disease, the less the diverse the microbiome. So as you can see here is the box plot showing that in the mild cases shown in blue are highly diverse as compared to the severe cases. So this led to the idea that the disease severity is linked to the microbiome, in their gut microbiome, and then that will tell a lot of other clinical phenotypes. So we were able to, based on these, we were able to phenotype the different girls, different groups, and we were able to provide them, dietary recognition or dietitian recommended different diets depending on their gut microbiome composition and things like that. It was a clinical study. And then I think they are following up the same study by another fellow or someone here in the Texas Hospital. Please bear with me. There are multiple studies involving different clinical aspects. The most recent one that we published, I think in May, is not a gut microbiome. It's in the cystic fibrosis microbiome. It's upper airway microbiome in children with the cystic fibrosis, which is very very I would say very deteriorating disease in children with they're very they have to go through a lot of distress and all kind of things. So what we wanted to provide some sort of ideas about how their upper airway microbiome looks, so that we can probably help them with their microbiome profiling and predict the disease severity or disease the outcomes using different algorithms. But this is the very basic study. We started as a pilot study and published back in this year in May, where we look at the 2 different populations of the Hispanic and the non-Hispanic. You may not have much familiarity with the Hispanic versus non-Hispanic. Here in the US, we have a huge population of Hispanics, particularly those coming from South America, or even they are here in Texas, for example. And then there are non-Hispanic population, particularly white American people. So what we look at the difference between the Hispanic and non-Hispanic, we found that the life expectancy. For example, those people with the CF, they have the decreased life expectancy and then earlier acquisition of the bacteria called Pseudomonas aeruginosa than the non-Hispanic white. And then we were asking questions if there are any racial or ethnic differences between these two groups, or we call them health disparity with regard to the airway microbiome. We enrolled the prospective observational study, the 2 to 10 years old with the CF center at the Texas Hospital. We collected the oropharyngeal, or basically oral swabs, and then we were able to sequence them using 16S-ribosomal RNA sequencing. What we found, interestingly enough, is that there was a higher incidence of the Pseudomonas aeruginosa in Hispanic CF microbiome samples as compared to the white non-Hispanic population. And there were one bacteria that stand out clearly was the Saccharomyces, the order label taxonomic rank, where you see the significantly higher abundance of this bacteria in Hispanic patient with cystic fibrosis. Taken together, all of these led to the ideas that using the microbiome in the clinical studies, you can differentiate the different disease type, whether it's whether it's a disease severe or mild or moderate, or whether it's the healthy controls or the disease status. Moving forward is next how does the microenvironmental factors fit into the microbiome and the disease? I think many of you are thinking about how does the diet plays a role, how the medication plays a role in terms of microbiome and disease. So this is just the figure that we in our upcoming book chapter, where we just showing that how the microbiome develops from newborn to early childhood, and when you get mature or elderly, and how these shaped in different ways. Microbiome diversity and complexity is you start the climax when you're adult, and you start decreasing when you become elderly. And there are multiple factors that can influence this microbiome. And the prominent one is the genetic factors, diet, and geography, stress, or drug, or environmental air quality and hygiene as well. So essentially, the focusing much more on the antibiotics and other medication, as you all know that it may suppress the commensal bacteria and may provide the fertile land for the pathogens to go to dominate, basically. And then based on the various studies, it has been shown that the diet has a significant role or relatively large impact on the biodiversity within the gut ecosystem. So then next, we also look at how does the diet play a role in certain clinical studies. For example, when I talk about the Rett syndrome study, we collected the gut and stool samples from these kids with the Rett syndrome and without Rett syndrome. What basically we found here is that those people with the Rett syndrome that were fed with formula or fed with the food means stable food, and their microbiome is different. So here in the labels or the figure legend C is that the observed OTUs, which is basically measuring the microbiome richness in the sample, and then the diversity, which is the feature diversity, measuring the diversity of the microbiome in particular samples. It clearly shows that those people who are eating the table foods have much more diverse microbiome compared to those taking the formula. The take home here in this bar, stack bar chart. So here are the relative evidence composition at the genus level between the two groups. Essentially, provided the foundation that the bacteria, which we call as a beneficial bacteria, Bifidobacterium, was increased in the formula consuming patients because their formula was supplied to the probiotics Bifidobacterium. Where there was not much the Bifidobacterium on the people consuming the table food. And then there was also differences between how much daily, fiber you intake daily. So there was some demarcation based on the American, Nutrition Society and others recommended. So there's some cutoff point where we make it the less than 4.1 gram per day versus the more than 4.1 gram per day. We're shown here in the principal coordinate plot, or we call it PQA plot. Shown here in the X and Y axis are the two major principal components, axes, which describes the highest variability in the data set. So this is the Bray-Curtis Again, when I say the alpha diversity and beta diversity, please keep in mind that the alpha diversity is within the sample diversity, when the beta diversity, for example, shown here in the PQA plot, is the diversity between the samples. Meaning these individual points shown here in the blue and red, the individual dot represents the sample. And then the points, the more closer the points, the more similar their microbiome is. So essentially those taking fibers in the high and the low rank, they have a different microbiome composition, is evidenced by the Permanova test of the statistical test. So the take home from this study is that there is a differences based on the dietary pattern, in particular the diseased person as well. Now, forget about the healthy control. But within even the diseased individuals, depending on their diet type, you would still see the differences in their microbiome composition, diversity, and then alpha and beta diversity as well. So this led to another idea that instead of looking at the microbiome, we also want to look at their metabolomes. So what we did basically here is that we ran the LCMS for the targeted metabolites. Shown here the GABA, which you may have probably heard. This is the bacterial end product of bacterial metabolism, and then other these metabolites like tyrosine and glutamate, tryptophan, and then glutamine. So and then based on their statistical value, you can see that by the way, the first A and B are the same for the GABA. One was with outlier and another was without outlier in the data set. So essentially showing that in the control means this is the healthy control. And then when you compare with the healthy control with the red individual, you will be able to see that the GABA concentration was higher in the controls as compared with the red individuals. And this is true for the gamma aminobutyric acid, but also true for the tyrosine. And there was a trend for the other amino acids called glutamate. So essentially showing that not only their microbiome, but also their functional microbiome, if you will, or what are the end products of their bacterial metabolism are also different between the healthy controls and then diseased individuals, as exemplified by the girls or women with the Rett syndrome. So moving forward, I don't know how long I have time, but I'm not sure. So, but let me check quickly. Okay, so essentially, this is I think the last study that I'll be talking much more about how the different disease and then microbiome plays a role or help in clinical diagnosis or help in management of the disease. Is one of them is this one is the effect of antibiotics in the children with otitis media, where your antibiotic exposure with regard to the nasopharyngeal microbiome. In this particular study, what we did is that, as we all know that there is a high use of antibiotics in pediatric outpatient clinic, not only in the developing countries like Nepal, but also in the US as well. They use tons of the antibiotics for these kids going through the otitis media, for example, or ear infection. There are some indications that even people prescribe antibiotics for at least 90 days before doing some tympanostomy or other eye-related surgery. Ear-related surgery, ear infection. So there is a high use of antibiotics in the field of the pediatric outpatient settings. So for this study, what we did is basically what we collected the rectal swabs from these kids who are undergoing tympanostomy, and also collected the nasopharyngeal samples for these children 1 to 6 years old. I think there are 50 kids in this group. And we essentially analyze the data using the 16S ribosomal RNA sequencing, and then analyze the data based on the alpha diversity, beta diversity, and then the relative abundance composition. So here in the figure A is the Bray-Curtis dissimilarity, which is a type of beta diversity indices that we use in the field of microbiome. In the x-axis and y-axis, there are axis 1 and axis 2, which represents the highest variability in the data set. So shown here, a group by the antibiotics, yes or no. You can see that there's a separation of these dots into the blue and the red ones, essentially suggesting that their microbiome composition is different depending on how long they are under antibiotics versus not. Similarly, for the figure B, shown here is the alpha diversity, as exemplified the Shannon index in the y-axis, and the number of antibiotics doses in the x-axis. You can see that there's a correlation. It's not significant, but it's sort of negative correlation, just to indicate that. And more importantly, when we look at the relative abundance composition, of these two different sample types. With the antibiotics or without antibiotics, you can see the spiking of these bacteria called Haemophilus, which is one of the otopathogens or causing ear infection in these children. So essentially supporting the fact that these otopathogens are highly dominant in those kids treated with antibiotics versus not. And the last piece of my talk would be focused on the clinical utility of microbiome or microbiome therapy, we call them, or some people call it fecal microbiome transplantation or fecal microbiome therapy. I was over the last, I think last 2 or 3 years, I was responsible for collecting the fecal samples from the donors and then, getting ready these fecal microbiome samples for transplantation to the real patients who are suffering from the Clostridium difficile infection. That was part of my clinical medicine training in the hospital. So what essentially in 2019, I believe, we published one paper with the toddler who was undergoing the heart transplantation and who was suffering from the recurrent C. diff infection. Essentially, there's always a high risk of the people who are receiving the soft organ transplantation. There's always a high risk of the Clostridioides difficile infection. This patient particularly had 5 episodes of the recurrent CDI in the last 6 months. And we use these 6-nest ribosome RNA-based techniques to profile his microbiome, basically. So here in the figure A and B is that in the y-axis is called the Shannon diversity index. In the x-axis there are different groups. One is the donor samples. As you can see in the red, the higher alpha diversity, and we also include a healthy control. They also have the very much closer to the donor samples. And then when I look at we can look at the kid who was undergone, the soft organ transplantation, who was suffering from the recurrent C. diff, the pre-FMT, their microbiome was very low, almost close to 1. And then post-FMT, we provided the fecal microbiome transplantation first and then second time. The first time, and then we collected these 2 samples, I think 4 months later. And then the second time we collected, I think 16 months later. So you can see that, after the first and the second FMT, that you can see the increase in these alpha diversity indices for this particular sample. So when you look at the beta diversity indices, you can see that the pre-FMT is here in the figure B and the top corner, I would say top left corner. And then whereas the post-FMT and then 1 and 2, they're very closer with the healthy control side. So essentially, it provided evidence that this fecal microbiome transplantation restored the microbiome diversity without any complication or without transplant complication. And it prevented, further recurrent C. diff infection in a follow-up for up to 4 years. So that was great help for the direct patients. And we have been doing this similar type of study in another study as well, which is ongoing with our collaborator here. So this is just an evidence showing that how the fecal microbiome transplant can work in these kids. But there are still the reports that are not working, but depending on these outcomes, give or take. But fecal microbiome transplantation is still a very feasible techniques. So the key takeaways from my talk today is that the microbiome dysbiosis has been implicated in various disease types. As I saw in one of these studies, that the catheter biofilm microbiome varies depending on the infection status in the neonates, suggesting that the microbiome can provide and differentiate between the healthy versus the disease status. In Rett syndrome, what this study, I've shown the data that the alpha diversity decreases with increased disease severity. Likewise, there is a racial disparity in the breast tumor microbiome of the two, US population. Similarly, the gut microbiome changes over the age, and then diet has a significant impact on the gut microbiome of the individuals with, for example, with Rett syndrome. And then antibiotics in the early stage of the disease are the significant disruptor of the nasopharyngeal microbiome. And finally, the fecal microbiome transplantation can restore your microbial diversity and can prevent from the recurrent C. diff infection. With that, I would like to acknowledge my, PhD university from my PhD, University of North Texas Health Science Center at Fort Worth, and my previous and current mentors and collaborator here at the Baylor College of Medicine and Texas Hospital. Also, I would like to thank the funding agencies and then, the study participants and their partners. And without further delay, I would like to, just to give an announcement that we are also organizing the 4th edition of the International Conference on Infectious Nanomedicine in December in Kathmandu. So the abstract submission deadline is August 31st. And this just to show you that we had a very well-renowned speaker in our previous editions, including 2 Nobel laureates. Back in 2012, we have a Professor Barry Marshall from Australia, including other renowned professors and scientists from all around the world, including those from for example, Geoffrey Smith from University of Cambridge, which is now the University of Oxford, and then Yuen Kwan Lee, which is sort of, I would say, the father of the gut microbiome in Asia. He was based in Singapore, and there are others, from Rob Bragg from South Africa, as well as in 2015, we had the privilege of having the Dr. Morshid from University of British Columbia, Canada, and unfortunately, Dr. Anand Chakraborty passed away because of the COVID-19. And then 2021, we had Nobel laureates Ed Ayona from Israel and Professor Peter Hortes from my college, Baylor College of Medicine. He's a very well-renowned clinical scientist around the world. And then, of course, you may have heard Jack Gilbert, who is a very well-known microbiome expert in the field of UC San Diego. So this year, we are also planning to have at least very reasonably well-seasoned scientists for the meeting. So with that, I would like to thank you for your attention for the entire time. And if you have any questions, please feel free to ask me.
Thank you so much, Dr. Thapa. If anyone have question, either you can type in the chat box or you can unmute and ask.
So, Santosh, here I sent a question to you. Your slide segment you 16 years put here the B1 B9
Give me 1 second.
B1 to B9.
Okay. Hold on a second. B1. Let's see.
Maybe third, fourth cycle.
Sorry.
Yeah, I think.
This one you're talking about?
यसमा विषय के थियो जस्तै होल बेस कन्सेप्ट हैन हामीले पनि यहाँनिर सिक्वेन्सिङ गर्दाखेरि फर्मुलेसन सुरु गर्छौं हैन
I can barely hear you. Can you please I don't know, it's coming the echo. Prozulji echo आइरा छ कि I'll take that. Okay, please go ahead.
मेरो क्वेशन के भन्दाखेरि जस्तै हामीले यो 1500 बेस पेयर हामी सिक्वेन्सिङ गर्छौं नि हैन टोटल हैन तर यहाँ भित्र पनि भेरिएबल रिजल्ट्स अफ B1 देखि B9
यस
हैन त्यो जस्तै कुनै दुईटा B1 B2 गरे पुग्छ कि B3 B4 गरे पुग्छ कि कुनै पाँचवटा कम्बिनेसन गरे पुग्छ कि होल गर्नु पर्ने खासमा
That's a very good question. It's cost, I mean, if you don't mind, I'll for all people. So the B1 to B9, these are the hypervariable regions. So the Illumina technology, Illumina MySeq sequencing, for example, has not yet developed the strategy for the full and 16S sequencing. They only have the parts. For example, you may choose B1 to B2, B3 to B4, B3 to B5, depending on your objectives. But many people have chosen either B4 or B3 to B5. But I will tell you that there's no consensus, no written things that you have to do this. It depending on the variable region of your choice, you may have a little bit different results. But if you can do the full and 16S, which the PackaBio does, the PackaBio is a company which has developed the tool, but not the Illumina. The Packa, if you use the full and 16S, and there are many papers coming out recently that are showing that if you do the full and 16S versus, for example, V4, you would have a higher resolution in your findings. But provided that you would have a smaller, fragment in the V4, for example, it would be much easier, much cheaper, and all sort of data analysis will be less complicated compared to the full and 16S. So, I mean, you can choose B3 to B5, B4 is the most widely used, I would say, but there is, like I said, I'm afraid to recommend anyone, but because the it depends on what is sample type, for example, whether it is the stool samples from humans or stool samples from other animals or the other environmental samples. So depending on objectives, you can choose any reasons, B3 to B5, B4, B1 to B2. These are the most widely used, but B6, B7 to B9 are less widely used. Less used, to be very honest with you.
This way,
So,
when it's sample or depending on this way.
sorry?
Sample or depending on this way.
Well, I mean, your sample, and a sample and then your objective. How high how higher you want to go resolution. If you want to go to the high resolution, you can do the full length 16S. But for example, even in the US, it's very difficult to run the B1 to B9, for example, unless you have Pacabio machine. And it's not cheaper compared to the particular variable region. For example, the Pacabio 1 sample for the full length 16S costs $90. And for the particular, let's say B4 region, in our center, we just cost $10 or even less than $10, depending on the number of samples. So
Thanks.
there are a lot of things, the resources wise and then objective wise. The more you cover, you'll get a better result. But choosing B3 to B4, B3 to B5, B4 or B1 to B3, there are not much differences. You may get different results, little bit slight differences between the B1 to B3 versus B3 to B5, but it's still almost the same. So, does it answer your question? Okay.
Thank you.
Yeah.
Anyone have any more questions?
Santosh sir.
Yes, please.
Here I have one technical question. It's related with the 16 RNA sequencing. So I've seen that you are using the gene specific primers using adapter with an adapter, I guess, instead with the tag. So is the way of that you do this sequencing? I don't know how do you do the 16 RNA sequencing.
This is the standard way. It's the standard way.
Okay.
In the first step, you extract the DNA, and then you amplify particular region of your interest, depending on the primers. Let's say if you choose the
DNA
B4
or
region,
RNA, sir?
sorry?
DNA or RNA?
It's the DNA.
DNA or RNA?
This is the DNA.
Okay.
This is the gene. It encodes for the
Okay.
This is the gene. It's the 16S RNA gene. But we do the DNA extraction, and from there you amplify a particular region, and then this is all DNA-based sequencing.
Oh, okay, I understood. So I also did I also did a lot of this kind of RNA to the DNA reverse transcription. So
you can do that.
But I have not done 16S RNA genes, so I'm just concerned about it. Just asking. Okay, thank you, sir. And my next question is that I've seen that the cystic fibrosis cases you have compared between the Hispanic and non-Hispanic.
Yes.
So in that case, you said that your report says that the cases in Hispanic, I think,
right.
The cases in Hispanic is with the cystic fibrosis has a higher, I think, Pseudomonas.
Yes.
Pseudomonas acquisition of higher occurrence of higher frequency of Pseudomonas aeruginosa infection. So what could be this the reason? I think I didn't. Maybe you have told.
Well, honestly, we don't know the exact answer, but we envision that, or we thought that because when we look at the time point, at what point the Pseudomonas aeruginosa was acquired, it was earlier in life than the other white people.
Okay, so is it
like
something that they lived outside USA and they later they migrated to USA, and then probably they might have some kind of.
We don't know because many of the kids that we fall into the Hispanic population are migrants, so we don't know.
Okay.
And then keep in mind that not only that side, we also collected the data from their household. For example, whether their household has some smokes, or where someone is smoking, or their household has some, they burn the fuels or something like that. and it was correlated with those, you know, the household status, the education, they're cleaning things, and all kind of, you know, the health parameters. So that was also different.
Okay, that makes sense. Okay, thank you, sir.
No problem. Any other questions? If not, please feel free to reach out to me. I didn't post my email there, but you can just Google me, wherever you can find, or you can even you know, the email the conference organizer in the conference. So yeah, please. I mean, this is the microbiome field I've been very much here and dear since over the last 10 years or so. So if you are planning, for example, the you know, the microbiome research over back in Nepal, please feel free to reach out to me. I may suggest some ideas depending on my experience in the designing experiments, or, you know, whether you want to implement what type of controls, what type of technical controls you need, you know, things like that, or what kind of, you know, the population you choose, or what are the things to consider, whether age or the, you know, gender matters, or, you know, or geography matters, things like that. So, because there are very few studies in Nepal in terms of microbiome. We are kind of collaborating with some of the people's try to work in some aspect of the you know, microbiome in Nepal as well. We are still in the infancy, but we are in that direction as well. So,
Santosh sir.
Yes.
Sorry. So just a quick question. Probably this will be beneficial to many of the audience who are attending now. So since microbiome is mostly related with the bioinformatics and the many, you know, the statistical analysis, probably if you mention some of the tools and the techniques that we often use in the microbiome sequencing analysis, probably that will be great, you know, the advice to the audience.
Absolutely. I mean, I didn't mention specific tools because that would be a different story. Because if the time permits, maybe I am hoping to give a sort of, you know, the standard microbiome practice or standard microbiome data analysis workshop in ICIDN if we have enough participants in Kathmandu. But coming back to your questions, there are multiple tools. One of the widely used, the microbiome application tool or microbiome data analysis tool, which I often deal with every day in daily basis, is the CHIME, which is a canon. You know, pronunciation is a different, but it stands with the QIIME, which is the Quantitative Insights in Microbial Ecology. That's the most widely used tool developed by the Rob Knights and his group, you know, in UC Boulder. Now it's in UC San Diego. And also the other platforms they developed by the Pat Sloss in University of Michigan is called the Mother, which is MOTHUR. And these are the two major softwares that have been used in the field, but there are others as well. For example, there are PhyloSeq, for example, that we use pretty often time. And there are other tools that have been widely used. But long story short, the CHIME is the, you know, sort of consortia, whether you can, you know, you can plug into all other software packages as well. So that is the major, you know, data analysis tool that we have been using in daily basis. That's for the 16S-based data analysis. But I also deal with the shotgun metagenomics data sets. For that, we use the tool called the Metaflan, and then Human, developed by the a group in Harvard, which is the Harvard Labs. They have a Galaxy, you know, platform if you want to do the online, and there are there are multiple online tools if you want to use, if you want to play with. For example, even the Baylor College of Medicine has our own online tool. And then there are other tools, for example, Microbiome Analyst, developed by the scientists down in Canada. There are Microbiome Analyst also does a very nice job in playing with the data set of microbiome, whether you can, you know, upload the data and then you get the output results. Similarly, you know, the Atima, which is our software in the Baylor College of Medicine, is also the online-based tool. There are multiple different tools. You can use them.
Thank you, sir. Actually, I have asked this question because this information could be beneficial to the audience. So yeah. I believe they will reach out to you if they want to learn and have skills on these techniques. Thank you, sir.
Absolutely, no problem.
Santosh, how do you do fecal microbial transformation?
Say again, please.
How do you do fecal microbial transplantation?
Well, basically what you do is like you collect the you first of all you recruit the donors depending on their healthy status, and then depending on their, you know, we screen for a bunch of different parasites, bunch of different, you know, the disease conditions, preliminary screening. And if the screening fits into everything, he has not traveled outside the US, for example, and then he doesn't have a, you know, any diarrheal disease or any GI disorder or any comorbidities, then you would assume as a, you know, potential donor. Then we get consent from the donor, and then we collect the stool samples from them. Then we process the stool. We basically, you know, remove these larger particles, and then we prepare the slurry of the stool. We have in-house technique for you know, processing these samples, and then we make the slurry of these fecal samples, and then put it in -80. And then when the clinician need it, then they will contact us, and then we'll provide them, and they will do transplantation. This is the, you know, the pretty standard procedure here. But much more recently, we are just kind of holding because of the FDA ban on these in-house preparation of fecal materials. But we are still purchasing from some companies over here in the US. So we just, you know, process them and then send it to the clinicians when the patient is ready, or when they are doing mostly they do the rectal transplantation mostly.
Thank you.
Yeah. Are there any questions?
Do we have any more question, or if not, we can wrap up this session?
Absolutely.
Okay, I don't think we have any more question. Thank you, Dr. Santosh Thapa, for this amazing presentation and such a well-informative session. And again, thank you all the audience today. And I think we're at the end of it. And
thanks for having me. I really appreciate you guys. Like I said, please reach out to me if you have any planning for the microbiome stuff, and then, or if you have any difficulties in the, you know, the analyzing the data or suggestion, please feel free to reach out to me at any time.
Okay, sure, sir.
Yep. Thanks a lot.
Thank you, sir. Thank you so much.
Bye.
Thank you, everyone. Bye-bye.
Bye.